You were right. I am probably an odd one out here, but if you work on scorpions, you eventually have to work on venom. And so here I give a little overview of our work on scorpions in the last 10 years, but I picked out, of course, the bits that relate most to venom to keep it interesting. So without further ado, uh, let's talk about scorpions, which is my favorite thing to do. I work at CBO in Portugal, already said that. And the interesting thing about scorpions is, I'm talking about the scorpion arsenal, I don't just mean the venom, but actually there are two systems in the scorpion arsenal. One is, of course, the pedipalps with the pincers in the front, and the other is the, well, what people call the tail. It's actually an extension of the body, the metasoma, which has the venomous stinger at the end. And these two systems, these functional complexes, overlap quite a bit in their function. So on one side, you have the pedipalps, which are used for incapacitating prey. The prey is just crushed with the pincers, but also the venom, of course, is used for incapacitating prey. Likewise, repelling predators, you can pinch a attacker and then make sure that he doesn't return, or you can sting him. And so these scorpions have these two systems and they overlap in function, which means that there could be some sort of trade-off happening between these two systems, right? And these systems are also used in other functions like mating and sensing, although they're used in different ways. So there's quite a lot of demand on these two functional complexes in the scorpions, and that's what we've been looking at for, well, over 10 years now. So if you look across the scorpion diversity, there are more than 2,700 species of scorpions recognized right now. You see that there's quite a lot of morphological diversity. They're fairly conservative. I mean, they haven't changed that much. But if you look, for instance, at the scorpion down here, can you see my laser pointer? I hope you can.
Yep, we can see it.
Perfect. So this one has very large pincers in the front. It looks like a crab's pincers. And then if you compare this one to this one over here, which has these very slim, elongate pincers with very long fingers, that's quite a different situation. If you look at the metasoma, the tail, this one is a normally proportioned metasoma. This one has a hugely well, really thick metasoma, same length roughly, but very thick. And then if you compare those two to this specimen up here, it has very elongate pedipalps cala. The pincers are very long, but the fingers are very short. And if you look at the metasoma in the back, it's very short and it's very thin. So if you look across all this selection of scorpions, you'll see different combinations of how these two weapon systems are morphologically. And how do we make sense of this mess, right? Well, fortunately, I have a very good PhD student, Pedro Lobo Coelho, who looked at this, and he looked at scorpion ecomorphs, and he measured a bunch of scorpions and their dimensions, body dimensions, but he also looked at the microhabitat descriptors. He just looked in the literature to see where these animals were found and how that environment was described. And from those two data sets, and these are 61 species covering most of the families of scorpions, to make an extremely complicated and long story very short, is that we found that scorpions actually adapt their morphology to their ecological surroundings, like most animals do. And you can identify 4 different ecomorphs. You have, for instance, here, the sand species, the Pseudomophilus species, the Pelophyllus species, here in black. Those are the ones that dig a lot. And they have 2 climbing ecomorphs. You have the Phytophilus species, which climb plants, and you have the Lithophilus species, which hide under rocks, live in rock cracks, and climb rocks. And here, the interesting thing about this was that the parts of the body that vary most with their ecological habitat are the walking legs, of course, because they interact with the environment the most directly, but also the pedipalps. So the pincers in the front vary a lot with ecology, and to a lesser extent, the metasoma, what we sometimes call the tail. So these body parts change with ecology, very interesting. Here's a few examples of what they look like. Each of these ecomorphs is the species to illustrate. And if you look at the scorpion tree of life, you see that these ecomorphs have evolved multiple times in multiple families. So every time a scorpion goes from one environment or niche to another, it actually adapts its body in a certain direction, looking more like a Lithophyllus species if it moves towards living on stone, for instance. So it has a strong phylogenetic signal. It's keeping some of its ancestral shape, but then it's forming that ancestral shape into an ecomorph, into the direction of that ecomorph across the tree of life. And that's great. That explains part of the diversity. What it doesn't explain is what they do with this. Why are they so different, right? So the function of these different body parts. And I promise you, I'll get to the venom in a moment. Hold on. So in a very old paper, we looked at the defensive behavior in scorpions. We did something very simple. We just held down the scorpion by their prosoma, by their head, essentially, for a few seconds, and then held down each of the pincers to the ground for a few seconds, and we saw their response. And if they responded, they could respond by responding with pinching defensively, they could respond by trying to sting, and they could do both at the same time. Now, if you look at this phylogeny here of the species that we tested, at the top, you see a very large clade. This is the Buthidae, which is the largest family of scorpions. Almost half the species of scorpions are Buthids. And this is also the family that contains almost all of the dangerously venomous species. And you see that they use their stinger mostly in defense. You see a lot of yellow, you see a lot of red, which means mostly stinger, sometimes aided by pinching with the pedipalps. In the other families, all the other families down here, you see that there's also some species that have prefer to use only the pedipalps, the pincers, to defend themselves. So there's quite a lot of variety in the behavioral use of these structures. So that's quite interesting. And one of the things when you do this kind of research and you poke at scorpions, you have to keep your fingers away, obviously. But if you do that, you get some really interesting results. So we know that they're used differently. We wanted to see if they perform differently. So here are two of the more extreme examples of the pincer morphology. On the left here, you see the death stalker scorpion, which has these really long, elongated pincers with long fingers. And on the right here, you see the very robust pedipalps of a, I think it's an Ophistotomus. Yes, it is. And so these are pretty much the extremes of the range. Do they do something differently in their performance? So we measured the bite force. And what we found is very interesting, because if you look at scorpions and you compare them to other groups of animals, of which we know bite force, and pinching and biting is functionally very similar. So we lumped that here. And if you correct that for body mass, you can compare them. And then you see that scorpions are among the hardest biters in the animal kingdom. Only crabs can bite harder, but they're also some of the weakest biters in the animal kingdom. So there's a gigantic range of bite force in scorpions. And that is, of course, begs the question, why? Why are they so extreme in their range? What functions are being traded off there? So that, there's the scorpions. So enter Yuri Simona, who is a PhD student and who looked at this in great detail during his PhD. He's almost finished. And what he found, one of the first things he found, that if you look at the closing speed of the pincers, actually the thin ones are faster and the big ones are stronger. I mean, it seems to make sense, but we needed to measure it. But the question is, yes, they're faster, but is that the function that is being selected for? So there may be a trade-off here between the venom. So if you can defend yourself with venom, maybe you don't need very strong pedipalps, right? Maybe you can be faster and then you don't need these extremely strong pedipalps. So we don't know what's going on there, but there seems to be some sort of trade-off between the pincers and the venom going on here. Now, finally, I will start talking about venom. Don't worry. It begins with getting the venom towards your target. Here are two species of scorpions. These two together are responsible for most deaths in North Africa and the Middle East. On the left, we have the Fattailed scorpion. And on the right, we have the Stalker scorpion. And although they're roughly similar in size, you see that the metasoma of the Fattailed scorpion is very big, and it's not fat. That is filled with muscle. They're extremely strong. They have an extremely strong metasoma, whereas the Deathstalker, much less so. And we were wondering, okay, what's going on here? Does this difference in morphology actually convey different function in getting the venom to the target? So what we did is we poked at scorpions, we made them angry. And we did this under the high-speed video camera. So if you poke them with a bit of wire, you can make them strike defensively and they won't hit anything. So you can actually see very nicely the trajectory that the scorpion makes with the stinger through the air. And if you record that from several different sides, then you can get the trajectory shape in 3D, which is very nice. Here you see one of these trajectory shapes. You see in color here is the velocity at each point. So it starts there in the blue and then speeds up very quickly. The red part is very fast and then it slows down around the curve. And we did that for many, for several different species. Some with a long metasoma, some with shorter metasomas, some with very thick metasomas. But we try to include these 6 here. These are very closely related. And what we find is not only that we can distinguish species based on how they strike, but also that longer metasomas are actually faster, not the thick ones. So we still haven't solved why we have these fat-tailed scorpions that have this extremely muscular metasoma. That is unclear because the ones that have a thin tail are much faster. Here's an example of one of these guys. This is a Deathstalker scorpion being bothered by me. And of course it is striking. And you can see the trajectory shape through space. This is of course slowed down quite considerably. This takes usually about 1/10 of a second for a strike. And what you notice is of course that the strike is very fast, which is very different from how they sting their prey. Because when a scorpion stings its prey to kill it, then it's holding the prey in the pedipalps and it very slowly looks with its stinger for a soft spot and then stings it. So that's a very slow and controlled behavior. It takes tens of seconds, whereas this takes 1/10 of a second, much, much faster. And when you strike this fast, you have a lot of momentum, a lot of velocity going on. So it's kind of like trying to stab somebody with a syringe, like here, this image from Pulp Fiction. And if you do that, you have, of course, the risk of bending your needle. And that would be a disaster for a scorpion, right? You don't want to break the needle. So the next paper, we tried to look at how scorpions defend themselves from this risk. And we wanted to know if the scorpions that use their stinger defensively more actually have a more robust, stronger stinger, so to avoid breaking it. So here is a 3D model of a scorpion stinger. It's called the telson. The sharp bit is called the aculeus, and the round bit that contains the venom glands is called the vesicle. And if we load this finite element model with a force at the tip, you can see in red, you can see the stress building up in the cuticle as the force gets larger, and then the force goes down again. It's quickly rotating it. But if you notice where the stress occurs, it mostly is here at the tip and here at the base of the aculeus where it meets the vesicle. And this is where you find that scorpions, if you encounter them in the field, they most often have their stinger broken off either here or just a bit of the tip. So these stress points seem to be quite valid. And when we did that across a very large number of scorpions all across the scorpion phylogeny. Again, we made these models, we loaded them at different directions, and we put that here, that data on the tree. The tree branches here are colored by, well, a risk category to humans. So we just took from a book, we took whether these scorpions are considered to be dangerous or not, and we use that as approximate for effective defensive venom. Of course, this is a terrible proxy for effective defensive venom. So if anybody of you venom experts has a good idea of what kind of data I can use to study this system better, I'm all ears. But what we found is, at least using this proxy, that the more venomous scorpions actually have less robust aculeae. They have longer and thinner stingers, and they are at much greater risk of breaking while they use them more defensively. And again, in defense, the forces are much larger than when they use it to kill their prey. So that is a conundrum. So yeah, if you have any interesting data we could put to get a better estimate of which stingers are used defensively against mammals, yep, give me a call. We'll leave that here, and we'll actually get into the actual venom itself, which again is used for incapacitating the prey, for repelling predators, and for mating as well. First, we wanted to see how scorpions decide how to use their venom. Now, we looked at some videos of, just nature shows where a mammal is attacking a scorpion. And what you see is that if a mammal attacks a scorpion, it usually attacks it repeatedly. So it tries to bite and lets go, tries to bite, let go, tries to bite, let go. So we simulated one of these mammalian attacks by having a jar with some membrane on it and just, placing it on the head of the scorpion, pissing it off, and causing it to sting. And we repeated that with a little pause 10 times in a row. So we tried to simulate a repeated attack by a mammal here. And every time we used a new cup so we could measure how much venom was expelled. So how do they decide to portion their venom during one of these attacks? Well, the results were very clear. They start out with a fairly large volume of venom on the first attack, and it gets less and less and less towards the end. And also the fraction of dry stings, so the number of times they sting and don't release any venom at all, increases with the attack number. So yeah, it seems that mammals have hit upon a very effective way of killing a scorpion. And the interesting thing is that the venom volume, if you sum up these venom volumes here, they're about between 5 and 10%, maybe 15% of the total volume that a scorpion has. So it's not because it's running out of venom that the venom volume is reduced. It is some other reason. So let's look at how scorpions inject their venom in a little bit more detail. How do you get their venom into the target? So we got it to the target, but now we need to get it into them. And of course, you use the stinger for that. And if you look inside the telson, there are 2 venom glands inside the scorpion telson. Here they're shown in green. The blue bit in there is the venom. So you see that there's a little bit of blue going forward towards the stinger, and that those are the 2 channels. So one from each of the venom glands. You have a channel going forward all the way to almost the tip of the stinger, and where you have small holes, one for each venom gland, where the venom gets expelled. And on the image on the right here, you see again a venom telson with the venom glands in blue, the gland itself in the venom in blue, the venom gland in green, and the pink stuff is the muscle that surrounds the venom glands and that contracts and increases the pressure and then pushes the venom out through those very, very long and narrow channels to the front. To see the performance of this system, and scorpions are fairly small, it's quite a different story from snakes, for instance. We built a tiny aquarium, and we filled that tiny aquarium with oil. And then we had one of the sides of the aquarium be a soft membrane. And we made our scorpions angry again. We have had lots of scorpions in our lab. We made them angry and made them sting the membrane. And what you see is something like this. I'll start the video. The arrow points to where you'll see the stinger go through the membrane and inject the venom into the oil. Here comes the stinger, and what you see is immediately two of these bubbles of venom forming, one for each side, so one for each gland. And because we recorded this with high-speed video in oil, means we can measure the diameter of these watery venom bubbles. And we know that, if you know the diameter of a sphere, of course, you know the volume. So we can measure the flow rate during this event quite easily. Now that we know the flow rate of several species, what we found is that they can vary their flow rate. So some have very low flow rates, less than 1 microliter per second. Some have very high, almost 10 microliters per second. And they can achieve the same volume. If you look, for instance, these bubbles and these green ones up here, roughly the same volume. They are achieved by either having a long duration, low flow rate, or a very short duration, high flow rate. So there seems to be a lot of control in how scorpions can deliver their venom. And we wanted to know a little bit more about the mechanics of this system. Again, if you look at how small these holes are, and this is quite a big species, and how long and thin these venom channels are here, you can imagine it takes quite a bit of pressure to push your venom through such small channels, especially if the venom is viscous. And extracting venom from scorpions, I found that it's quite viscous. So what we did to study the viscosity of the venom, getting us into the very fun field of venom biomechanics, is we extracted the venom of a scorpion into a glass tube that you see here. And so by extracting it electrically, we get all the venom out of the venom glands. And the first venom that comes out is actually very transparent. You can see here at the top, you can see the millimeter paper behind it, right through the venom. It's transparent. Then comes the opalescent venom. It's more proteins in there and other rubbish, and you can hardly see the lines anymore. And at the bottom, we have the very, very high protein venom, which is so dense that you can't even see through it anymore. Now, a classical way of measuring viscosity is by dropping a ball through a fluid. So what we did here in this case, it's a very small tube of venom. We dropped a 100 micrometer stainless steel ball through this venom. You can see it's sitting here, right here at the top. And if you do the same for a tube of water, what you get is something like this. First, the ball speeds up, and then it gets a stable velocity. That's the terminal velocity when gravity and the friction on the ball are in balance, right? Terminal velocity. So it stays the same speed. But if you do this in venom, and watch the ball here, because I'm going to start this little video. I hope you can see it on your screens. There goes the ball. I'll follow it along, falling through the venom, becoming less visible at the bottom there. Right, oh, here it goes again. And so this way you can get an estimate of the viscosity. But what we found is if you do that, you get a graph like this. So you get the initial speed up of the ball as it accelerates down due to gravity, and then it starts slowing down continuously. And then here the tracking algorithm loses the ball because the venom is too dense to see through. But you can clearly see that there is a decline in speed through the venom, which indicates that the viscosity is getting higher and higher as you go from pre-venom through opalescent venom to this high protein venom. And that means that the more venom is already expelled, the harder it will be for a scorpion to push more venom through these tiny little channels. So this may be one mechanism that may explain part of what we saw in the differences in injection velocity, but of course they may also have muscle control. So that's a very interesting field to continue to do. But I've kept you away from your molecules for far too long. So let's talk about those. We also did some venom transcriptomics because we want to compare, the composition of venom across species, and especially in an ecological context, of course. We did this the classical way. You kill your scorpion, you cut off the venom gland, you homogenize it, you extract RNA, and then you have a nice picture of the venom composition. We did that for 6 species here. And 4 of these are Buffet species, and then there are 2 other families represented here. And what you see is that the Buffets are more alike than these 2 non-Buffets, and you get all these lovely categories of venoms. The nice thing about doing 6 species in one go is that, of course, you have the same analysis methods, pipeline, and everything, so you can really compare them rather than having single transcriptomes published one by one, where it would be very difficult to compare really the venom of these species. So, but we thought, well, can we do better and not kill our lovely scorpions? So we tried a new method where we milked the scorpions at multiple time points. What we did is we extracted all the venom from the venom gland and then waited a certain amount of time and extracted all the venom again. And from that second milking, we extracted the RNA and we sequenced it. And it turns out that that works quite well. So here are our results. So this is, first we extracted the venom completely. We waited 2 days, extracted it again, and then we have this venom profile. Then we waited for several weeks. Then we extracted the venom again, waited for 4 days, and then we get this venom profile, where you can see that here at day 2, not all the toxin, not very many toxin transcripts are found, whereas in day 4 you get a lot more venom toxin transcripts in the transcriptome. Of course, we had to check against the classical way, so we waited another couple of weeks, extracted venom, waited 5 days, and then killed our specimen, and then extracted the RNA from the gland, and that looks very much like the 4-day venom transcriptome. So, and actually all the RNA was quite high quality, even in the venom. So you can actually now get multiple time points from the same specimen. So you could, for instance, feed it something else and see if the gene expression changes, or in different parts of the year, or things like that, all in the same specimen, which is of course a valuable thing to do, because otherwise you have individual variation to work with. Now, the reason this works for scorpions and it has never worked for snakes, we think, is because they have apocrine venom glands, right? So the cells bud off these vesicles that contain the product, but also contain a lot of cytoplasm, which contains messenger RNA, and that's, we think, what we're sequencing. And scorpions are not the only ones with this type of gland. There's some spiders, hymenopterans, and the platypus have this type of gland. We think it may also work for holocrine glands, where the entire cell lumen gets the entire cell contents gets released into the venom gland lumen, so containing the entire, even DNA of the cell. So that may work for things like cone snails or spiders or teleost fish. But we don't think it will work for merocrine excretion, because there you get a pure product vesicle released into the lumen without any cytoplasm. So it won't work for squamates, and people have tried many times for snakes, and actually sequencing venom RNA in snakes doesn't work. We know that. So in the end, you finally got some little bit of venom talk, but let me just run through some conclusions here. In the morphological diversity of scorpions, you see a correlation with phylogeny and ecology. So a lot of the diversity is explained by these 2 factors. Pincer diversity is definitely for force. You see these really big pincers, they are built for force. I didn't show another paper that showed a different line of evidence for that. And maybe they're built for speed. We don't know yet, but once Yuri Simonov publishes his paper about that, maybe we'll know more. So watch that space. And the metasoma function of these really strong muscular tails, we don't know what they're for. The stinger diversity, again, we have beautiful models, but we don't know exactly what venom components to compare them to understand how venom relates to the morphology of the stinger. And it seems that venom flow may be limited by increasing viscosity, but this is an interesting avenue to pursue that we definitely will pursue. And of course, last, you can get RNA from scorpion venom. Yay, and maybe your favorite species. Of course, this was some venom work in the last 10 years with all these wonderful people that helped me. And of course, I have to thank you for listening to me so far. I imagine you're looking at me like this. So I would love to hear from you. Thank you.
Thank you, Arie. That was lovely. And it's nice that you referenced some of the great movies that are around. These are two of my favorites. I mean, obviously, Pulp Fiction and The Big Lebowski have to be watched regularly every couple of years to not forget them. So people, this is your opportunity to ask Arie questions or share comments that you may have. If you don't want to do it on the screen, you can also type it into the chat. So let me see. There are some questions in the chat. Sebastian Duterte says, very interesting talk, Arie. Could you expand a bit on the RNA extraction directly from venom? What procedure do you use? Is it the same as tissue extraction?
It is. Yes, it's quite simply the same. And yeah, there's just so much RNA in there that it works fine.
Because yeah, his second comment is that he tried it with cone snails without success. I was thinking though, I know that for some species with the apocrine secretions of venom, these also have extra cellular vesicles in their venom, these little membrane-bound compartments. It's been shown for some spiders, it's been shown for hymenopterans, but it has also been shown for some species of rattlesnake, where they have small extracellular vesicles in the venom floating around, which have a different composition. They seem to be enriched for certain venom toxins. So it might still be possible that if you have extracellular vesicles, tiny chunks of cytoplasm with RNA are there. So, but this may vary, I guess, for different lineages, or the quantities are small, or maybe these vesicles are formed in such a specific way it's only enriched in proteins. But anyway, yeah, fascinating stuff.
Absolutely, and I think you will be able to find some RNA in there, but much lower quantities. Yeah, so maybe you'll have to amplify it some way. But I've so far only spoken to frustrated snake people about this.
And in this case, also a frustrated cone snail worker.
Yes, yeah, didn't know that. Thanks, Sebastian.
Mick Weber has a question. He says, really interesting talk. Thanks, Arie. Some excellent areas of research. He asks, can scorpions independently express venom from either channel, or are the muscles paired to only release the venom in like simultaneously?
In the high-speed video of the venom bubbles forming, we always see two equal bubbles. So it doesn't look like they have any separate control. And also the muscles that do most of the pushing, you can actually see it through the cuticle. When you extract the venom, there it becomes wider and it pushes the glands to the side walls. So more of the pushing is come from the inside, and it will be very hard to separate that between the glands. Okay. So probably both at the same time, always.
Yeah, that's interesting. I guess that's the same with snakes, I imagine, as well. I don't know.
Oh, I don't know. That'll be interesting.
I think I know only of one paper where they showed at least there seemed to be differential expression of the toxin profile between left and right. But I think that's something I wonder if people have ever looked at. You usually milk the snake in a normal way with both fangs going into your receptacle. So that's interesting too. Any other questions, people? Or as you think about these, I was wondering, since the pre-venom in scorpions is used for defense primarily or often, right? I mean, it's also the least viscous bit of the venom. I was wondering, are some scorpions able to spray their venom in defense?
Yes, yes. You have Parabuthus, for instance. That's a whole genus of scorpions, some of which spray, some don't. But actually, if you poke around with scorpions for long enough, you find out that all scorpions can spray if you make them angry enough. But the ones that really behaviorally spray, even when you just blow on them or approach them, those are Parabuthus. They spray their venom over quite a distance.
Oh,
And
wow.
those would be interesting to look at the viscosity, of course. We've looked at the viscosity of spitting cobras already. We didn't find any differences between spitting and non-spitting cobras.
Yeah. But there, of course, in the spitting cobra story, there are differences in bioactivities and composition related to it in spitters and non-spitters, because upon surface contact, if you want to quickly induce deterrent pain, you need to perhaps have certain adaptations. So that sort of conceptual framework can be transferred into scorpions as well.
Definitely, and we're already collaborating on such a study. We have already extracted lots of Parabuthus venoms, and they're now being analyzed.
Rock and roll. This is very exciting.
Yeah.
One of the mysteries you tried to answer, but you didn't really, of course, is why the fat-tailed scorpions have fat tails.
I don't know. I suspect it may be intrasexual competition.
In what kind of sense? Do they sting each other, or more like non-stinging fighting?
Yeah, so they fight. They will grab each other, try to grab each other, and push with the stingers. But we will need to do behavioral experiments to see if this is going on, and if the ones that can push with the tails and the hardest actually is the winner. So that is a nice little master project waiting to happen, I'd say.
Excellent. You almost make me want to work on scorpions.
Almost.
Anybody else some comments or questions for Arie? Well, if not, I thank you so much for joining us today. There was a really interesting talk. It's nice to see the diversity of approaches you and your colleagues take in studying these creatures. It's really stimulating. Everybody else, thank you for joining us this afternoon. Spread the word once we've put this video up on the EUVEN network YouTube channel. Everybody else who missed it can watch it. So that leaves me only to say thanks all of you. I hope you have a good rest of the week. I hope you have a good holiday in the summer, and I hope to see you guys back when the university starts again in September.
All right. Thank you very much. Thanks everybody for listening.
All right. Tot ziens.
Bye bye.